students, now we are going to take a next part of principle of inheritance and variation. In this topic today we are going to study about the blood groups. As we all know that human being has multiple blood groups like A, B, type A be there, type B will be there, type AB is there, and type O is there. So how this blood group can be identified, or with the help of genetic basis how we can find out what blood group is going to we are find in the offspring. So it is first we will know how to represent it. How it is represented? It is represented denoted by capital I, and if it is having antigen A, then A is mentioned. It is clearly given in the paragraph. You can find it out very well. If antigen B is present, then IB. If in any case there is no antigen, then small I is represented. This you can see in this table itself. If parent first is having, in the first column you can see, if parent first is having IA, that means it is having antigen A, and if parent 2 is also having the same antigen I, and that is A, then the offspring will be having both the antigens IA, then the blood group will be A. In second case you can see parent 1 is having antigen A, but parent 2 is having antigen B, IB, then the genotype of the offspring will be IA and IB. Then both the antigens are there, then the blood group will be AB. Understood? In the third case you can see IA and parent 2 is having I, means there is no antigen. So genotype will be IAI. That means only antigen A is there. So blood group will be A. In the last case you will see both the parents having I. That means there is no antigen into it. So genotype will be double I, small I. Then what will the blood group? Obviously O. So I think you might have understood how we can find out the blood group of an offspring. Now here is a table which is showing the possible blood groups of children from known blood group of the parent. When the blood group of a parent is known, so it is basically same thing which I have told you in a previous slide, that is blood group of parents is A and A. The possibilities of the children will be A and O. Why O is there? In case the parents are having a recessive O which can be dominant in the F2 generation, that's why it is O. So chances of O will be there. The impossibility will be, as there is no antigen B, so hence there will be it is impossible that the blood group will be B or AB. In the same case, in the case of second case, you see parents are having blood group A and B. Both are the dominant antigens. So blood group can be A, blood group can be B, blood group can be AB, and blood group can also be O. But impossibilities are none. Apart from that, it will be having some blood group. All groups are being included. So this is how the blood group of a parent decide the blood group of a child. I think you might have understood this. Now moving to the next slide. Now, what is polygenic trait and quantitative trait? When there is a continuous variation is due to the additive effects of two or more gene on a trait. If certain traits are there, certain gene are there which are producing impact on a certain traits. As we are studying traits, we are studying inheritance, so we are understanding how the effect of dominant or recessive traits takes place or affects the other alleles or gene. When two or more traits affect any certain gene, then the condition is known as polygenic trait. Okay, or it is can affect in a case of inheritance, then it is a polygenic inheritance. Okay, now this can be understood with the help of this checkerboard where three set of traits being used. How many traits? 3 tests. Now just watch the table carefully. White, very light. Okay, so the we can say alleles used are small a, small b, and small g. If we form a gamete of that particular trait, then we will be taking a single chromosome, that is small a, b, c. Okay, this is now second trait is black. What it is having? Capital A, capital B, and capital C. These are the parents. So what is the gamete of it? A, B and C. So if we produce F1 generation, that means gamete of parent will produce with a cross with the gamete of parent 2, and it will form the F1 generation. So what will be F1 generation? That is capital A, capital B, small v, capital C, small c. Now if we do a checkerboard, if we made this cross, so cross as I told you in a diamond cross, same happens with the that is this cross, where our quantitative inheritance of a skin color is being taken, and you will find that where whatever the place the dominant trait A, B or C is there, it will be showing clearly the dark color, as it stands for dark color. If there is no dominant trait or dominant character over there, then it will be showing white color, or if the traits are being mixed, then it will be showing intermediate color. So this is a polygenic inheritance. Okay. Now we have a pleiotrophs. What is pleiotrophs? Pleiotrophs is a responsibility of a single gene for more than one phenotypic effect, often seemingly unrelated, is known as pleiotrophs. It simply means a single gene for a tallness or shortness, for white color or black color of eye, whatever the trait it may be, it is showing continuously effect more than one phenotypic effect. But it means whatever the physical characters it is going to affect, like the amount of melanin in the body, it affects the skin color, eye color, hair color. So one single cell character trait can affect the other phenotypic things as well. For example, it is given over here. An interesting example of pleiotrophy of hereditary disease is sickle cell anemia, genetic disorder. This is a genetic disorder which can affect the inheritance. Okay. Now, pedigree analysis. This is very much important because it used to come in the board. Pedigree analysis. Pedigree analysis is a record of the inheritance of a trait in several generations. For example, if we are going to study any parent, what are the genetical disease or good traits in particular parent, so we will study its pedigree analysis we will do, and we will study its inheritance procedure, how things are being inherited to the certain generation. The person from whom case history of a pedigree starts, from where the point of start of the male parent, from where it is going to start, that parent is known as proband. Okay, and means the point from where pedigree analysis starts is known as proband. If that is a male, then it is a proposity, then it is known as proposity. And if it is a female, if we are starting from a female, then proposita. So how it is done? This is a symbolic used in a human pedigree. If it is male, then we will make a square, female then circle, sex unspecified hai, then we will the symbol. If the individual is having disease, then if the mating is being done, and mating between the relatives, if they are married within the relatives, then this sort of symbol is being used, and parents of the children below, order of the birth, left, right. So these are the symbols which we use, and we can make a pedigree chart. So if you have understood this, I am expecting that in your notes you will make your family pedigree chart. Family pedigree chart from means grandfather. You can pick from your grandfather, and then you can make a pedigree chart using these symbols. So hope you have understood this. Do prepare the notes properly and submit notes on time. Thank you very much.